Hello, it's Iago San Martin Villar. I would like to share with you some topics focused in within and between behavioral variability in ants that I have studied with Raphael Janson. Behavioral variability allows individuals to perform different tasks, and thus it is considered as a necessary capability for division of labor in social insects. Behavioral variability depends on several factors as the developmental time, gene expression, neural connections, seasonality, colony number, and phenotype. In this presentation, I'm going to focus on how behavioral variability is affected by social context and how differ between castes. The social context perceived might depend on the colony size. The increase of colony size produces an increase in task specialization. Individuals are more generalist at low colony sizes and more specialist at high colony sizes. The increase of colony size also produces a reduction in the ratio between brood and workers. There are more brood than workers at low colony sizes and more workers than brood at high colony sizes. According to that, we expected that individuals in the early stage, in this case larvae, will perceive the colony status by the number of interactions with adults and by the specialization of these adults. In low-size colonies, larvae might perceive the task changes performed by a low number of generalist adult sisters due to the high fluctuation on their care. Whereas in high-size colonies, larvae might perceive the task specialization of their adult sisters due to the low fluctuation on care. We hypothesize that early experiences like this could shape the future adult behavior as a response to prepare individuals to future conditions and colony necessities. We replicated two treatments with different brood-worker ratio to observe if differences in social context perceived across larval development affected individual behavior when emerged. We performed a total of 90 experimental groups from 3 Lasius niger colonies. Hereafter, I will refer to these treatments as high and low brood treatments. As predicted, larvae receive more contact by adult sisters in low brood treatments. Here, we can see how the number of workers in contact with brood increase in low brood treatments for the 3 colonies. We measure the distance between eyes as a proxy of body size and observed how adult emerged from low brood treatments showed larger body size, even if differences between treatments depended on the experimental colonies. In addition, larvae reared in low brood treatments had a longer development, spending in general more time as larvae. We performed behavioral tests with emerged individuals reared in the 2 treatments. We tracked individuals' activity for 1 hour in 10 daily tests. To analyze between individuals' variability, we compared the mean time that individuals spend moving between treatments and the repeatability of their activity. It means the proportion of total variance that can be attributed to variation between individuals. To analyze within individuals' variability, we compare their plasticity. It means the extent of change as a function of time, and their predictability. It means the variance from an estimated regression. We expected that individuals reared in high brood ratio would perform higher scores in the 4 behavioral dimensions measured. However, we find no differences in behavioral variability between workers emerged in both treatments. Workers differed in plasticity. It means each of them adapted their behavior following a different pattern. But the variability observed was similar between treatments. The high plasticity of workers and its independence from early social experiences make us questioning if this variability was inherent in workers because they must perform different tasks across their lives. Therefore, we hypothesize that phenotypes developing less tasks, for instance queens, might show similar plasticity between conspecifics of the same caste. In this case, we tested workers' and queens' activity across 10-minute tests for 5 consecutive days. We selected the Linepithema humili species as a model due to the high number of queens present on each colony. In contrast to our expectations, queens differed in plasticity, and in contrast with the observed in Lasius niger, Linepithema humili workers performed a similar pattern of activity. It means workers did not differ in plasticity. To sum up, we observed how social context perceived at early stages modified larval development, but not their behavior. This could be explained if behavioral variability is canalized. It means if it is fixed under certain thresholds. In our experiment, larvae reared in high brood treatments were less assisted by their adult sisters, and probably they receive less resources than the sisters reared in the low brood treatment, which could grow more due to the higher resources received. Therefore, larvae reared in high brood treatments had small size when adults, but also a faster development as it happens in foundation colonies. The increase of body size could not be linked with an increase of their behavioral capabilities, and thus, individuals showed similar variability independently of their early experiences. We also saw how Linepithema humili workers showed similar plasticity. Workers with a limited threshold of behavioral variability could need less complex neural circuits, which could involve a reduction of cost for colony production. This result was not present in Lasius niger, which also showed a decrease in their activity through the time, instead the increase performed by Linepithema humili. These kind of differences could be related with the colonization capacity of Linepithema humili, but it should be noted that these differences could be due to the age. We tested callow workers in Lasius niger, while we tested adult workers in Linepithema humili. In contrast to workers, Linepithema humili queens differed in plasticity. Following the previous argument, colonies could afford in costs related with cognitive skills by allowing behavioral variability only in queens, which are a low number in comparison with workers. Differences in plasticity in queens could increase the capability of adaptation of these invasive species by allowing the colonization of different habitats. I hope you have enjoyed the presentation. I would like to thank your attention and interest. I also thank Isettos Emrede for the opportunity to participate in this interesting workshop. The experiments shown were performed thanks to the funding of the Galician government and to the work performed by our colleague Eneko Sata and Gérard Latil. Thanks for watching.